I'm very happy to introduce Dr. Kert Viele, who is joining us from Berry Consultants today. So Kert is the Director of Research and Senior Statistical Scientist at Berry Consultants. That is in Austin, Texas, in case you're not familiar. He has been in that role since 2019, though he joined Berry back in 2010. For those of you who are unfamiliar, Berry Consultants is a statistical consulting company specializing in Bayesian approaches to biomedical research, especially adaptive clinical trial designs. He is also affiliated as a professor at the University of Kentucky. He was an assistant professor there from 96 to 02, associate from 02 to 10, and now the professor appointment. If you looked at his CV, Kurt's got a lot of accomplishments and activities going on, so I'll just highlight a couple of things. He's a participant in multiple FDA, CTTI, and Bayesian analysis working groups. While at Kentucky, he was a recipient of the Provost Teaching Award. Congratulations on that. And happily, last year, he was inaugurated as a fellow of the American Statistical Association. Kert and I share a connection. For those of you that may know, I was a student at the University of Kentucky. That's where I got my master's degree. And Kert was one of my professors while I was there. He taught the Theory of Statistical Inference course out of Casella and Berger. Second edition, in case you're familiar with that one. I still have the book. It's held together by duct tape. I struggled in that course, and Kert was exceedingly patient with me during my time there at UK, with extra tutorials, encouragement, and dinner at TGI Fridays pretty frequently to help work through not just issues in the classroom, but issues as a graduate student and living in Lexington. He truly is a gifted educator, so it's no surprise that he won that teaching award, but he's also a great person to be spreading the gospel about Bayesian adaptive designs. I invited Kert to share some of his experiences specifically from his time with Berry and as someone who has worked both in and out of academia. The title of his talk is, well, I had case studies, but Advanced Analytics, Bayesian Adaptive Clinical Trials. And I will turn it over to Kert. So please join me in welcoming Kert Viele.
Thank you all for having me. It's great to be, I work from home, so anytime I'm allowed around near people, it's a very great experience for me. So I want to talk about really 3 case studies in adaptive Bayesian trials that are somewhat cutting edge. Hopefully they're relevant to your lives, that come from the COVID epidemic. So these are trials that have influenced how people are treated for COVID today. And I've tried to, in these slides, end each little case study with a description of active research areas that essentially there's lots of problems in here. This is going to get described as, oh, this trial is really, really cool, and it does a lot of cool things. At the end of it, it's going to be like, it's cutting edge new, and there's lots of things about this we don't fully understand. So here's what they are, and if any of you would like to work on them, we would be delighted in any way to have these problems solved, and are happy to participate or comment or otherwise just watch you achieve stuff as you choose. So the modern clinical trial, a lot of you may know a lot of the stuff I'm talking about, but it dates back to 1948. There's a trial in streptomycin. There were lots of clinical investigations before 1948. And you can see a lot of the pieces of a modern clinical trial come together. Stuff like doing one-to-one allocation, and stuff like making sure that data is handled well, and there's objectivity in making assessments. All of that stuff happens. What really happens in 1948 is randomization. So there's a big trial that is considered like the almost first clinical trial in 1946, but what they did is they allocated patients where you get treatment A, you get treatment B. You get A, you get B, you get A, you get B, and they did that, but everything else looked like a modern clinical trial. In 1948, they decided to flip a coin. So very exciting moment. Statisticians have flipped a coin. Okay, so anyway, the nice thing about this modern clinical trial is it is very scientifically robust to answering a simple scientific question. Does streptomycin work? So we've got a whole bunch of people who got streptomycin, we got a whole bunch of people who got a placebo. We can get the question of, do people do better on streptomycin? It is very large. It is also very, very expensive to run. These require, I don't know how much the 1948 trial costs, but nowadays a trial like this would cost many millions of dollars, potentially hundreds of millions of dollars, depending on how big it is. And the actual indication where streptomycin is being used, I think it's tuberculosis trial. Okay. The other thing that is, while this is a very robust answer to a single simple question, there are some quirks about it, which I'm going to get into, where it is not robust at all. And so there's lots of situations where people get to the end of a trial and they've spent $250 million and their thought is, boy, I wish I'd run a different trial. And so I want to talk about some of where those might come up, and really what adaptive trials are aimed at is trying to minimize that risk. So I'll get to there in a moment. One thing I often, parts of this talk I do for applied audiences as well, and I have a lot of people who go, well, you know, this is it. This is the clinical trial. This is the gold standard. We should never change this. There's literally nothing about our lives that hasn't changed since 1948. You know, I actually showed pictures and other slides of this of a 1948 operating room and a computer from 1948. I thought there would not be a cell phone. There apparently were cell phones in 1948. If you go onto YouTube, you can actually see ads for this, which I was fascinated to find out. They're not very mobile, but they exist. But anyway, all of this, you know, we can imagine that the clinical trial should be advancing in ways as well. All right. So a lot of times when people hear adaptive trials, they often think of things like group sequential designs. And I don't know all of your all's experience with adaptive trials, but I have a lot of people come and talk to me. And essentially what they've heard is if they do something adaptive, they can run a smaller trial. If I run a group sequential design, I can have a smaller sample size. And one of the things I want to talk about today is that's really not where my life exists. What I want to do most of the time with an adaptive trial is not just try to make things shorter. I want to either get an answer to a more complex question, or I want to get a more robust answer to what I have going right now. And one of the ideas behind adaptive is think of all the things when you design a trial that you don't know. And so, I mean, there's tons of this. You can imagine, you know, I run a phase 2 trial, everybody comes up and it's like, here's the data from my past trial. I now want to run a big phase 3. And so a lot of times phase 2s, you know, it's not like you get a p-value of less than 0.025. So they come in, they've got a trend, everything's looking great. But they might end up with a confidence interval like this -1 to 5, my point estimate of 2 points. So tell me what my sample size needs to be. If I'm not going to run an adaptive trial, I have to pick something advanced. I pick a number. Do I pick a big number? Do I pick a small number? The trial that you would design for a 2-point effect has a very different sample size from the trial you would for a 1-point effect, and a very different one than the sample size you'd have for a 4-point effect. So given that fact, this is one of the reasons I'm saying that a fixed trial comes with risks. So if I pick a big trial, I'm going to end up in the situation where, okay, it's going to be adequately powered for basically everything in this confidence interval. But of course, it might take several years longer than it otherwise might need to. And in no situation in the world is there only one drug to be investigated. So all of this should be taken into account with the idea of every drug that we spend extra time on patients on now is another drug that got a sample size of 0. On contrast, if I pick a small sample size, well, that's great. I can run a small trial, but it's quite possible the trial may be underpowered. At the end of the day, I didn't answer my question. That's a bad time as well. So that's a simple one where we run into this level of uncertainty. often run into, this is a case study I'm going to go through, treatments for Ebola. There were lots of, during the Ebola epidemic, there were lots of different drugs that were suggested, and there were lots of different combinations of those drugs. And so the question is, if I have, 10 different combinations that I might try, how do I investigate those? Do I investigate them one at a time? Do I investigate the combinations first and then worry about the singles? Do I do one drug and then potentially, if it works, I try combinations of it? If it doesn't work, I try combinations of it. You can imagine all of these situations where, if I have lots of different possible drugs, what do I give first? How do I explore this kind of space? And a standard clinical trial would often say, pick one, see if it works. If it doesn't, pick another, see if it works. That's not an efficient process for that, which we'll talk about when we get there. There's lots of situations where people, when they're deciding on a trial, they don't know who to enroll. So we often have a situation where people come to us and go, I think this works in everybody, but it's possible it may only work in people who have the severe form of this disease. Or you need to have a certain biomarker, you need to have a certain gene, but I don't know this. And so you can imagine the issue where if I run a trial that enrolls everybody, well, if it actually doesn't work in some people, you've diluted your treatment effect. You make it harder to see the people that it actually does work in. In contrast, if I run the trial in only a small set of people where I'm most optimistic, well, if it works, I might be able to demonstrate that, but it's a trial that takes longer to enroll, and I may miss the fact that this drug benefits other people as well. So there's lots of all this kind of hedging that you're going on. When I say that a standard trial, it answers a specific question, one of the things I'm getting at here is that question itself is limiting for us. And I want to say I'm using the adaptive trials here, not to just save sample size, that's not what I'm aiming for here, but to try to answer these more complex forms of the question. And then the last example, things like factorial designs, which I will get to in due course. Okay, so I do Bayesian trials. I'm going to take a survey. Can I take a survey? Go for it. All right, how many people, if you were to classify yourself as Bayesian or frequentist, would go with Bayesian? All right, okay, this is a weak Bayesian. So anyway, I'm quickly going, who's on my committee? Is this going to go well? All right, I assume everybody else is frequentist?
I like to count things.
You do like to count things? Everybody can count things as well. So that makes sense. So I am not a hardcore person in either direction. You can find all kinds of stuff where people will tell you, oh, if you're a Bayesian, this is utopia. If you do frequentist, it's the apocalypse, or vice versa. That is not me. That said, you will find that we do most of these from a Bayesian perspective. There are reasons for this. Our kinds of trials, they often involve incorporating multiple sources of information. Bayesian has a nice convenient mechanism for doing that, incorporating one, and I know there are frequentist analogs to it, but this is just what we found this to be easier, bringing in information from prior studies, longitudinal data, all of these different kinds of pieces, putting them together. We often find this to be easier from a Bayesian perspective. I'm not saying it is, because you should all obviously keep in mind, I was trained as a Bayesian and not a frequentist. So in that sense, this could be as much my ignorance as the actual truth. This is a big one, and I do think the Bayesian is a little bit more on the, I'm not sure there is a frequentist analog. A lot of adaptations involve predicting the future. So if I'm partway through a trial and I want to ask the question, should I just stop this trial for futility? Should I give up? One of the ways this is often phrased is, do I think I have a chance to win this trial? So if I'm at a sample size of 100, the maximal sample size is 400. Do I think I'm going to win at 400 if I continue this? And in order to answer that question, there's two levels of uncertainty. There's one level of uncertainty that I've got future data. So data is random, binomial, normal distribution, whatever it happens to be. And in addition, the other level of uncertainty is the fact that at a sample size of 100, I don't know the truth of the drug. So I don't know whether it actually works or not. So if you've ever heard things like conditional power, predictive power, what a conditional power will often do is make an assumption about what the truth of the drug is and predict that forward. The Bayesian methods account more for that uncertainty in the parameter. And so that's one of the reasons that they're liked in this case. Again, I'm not adamant one way or the other on this, but especially for small sample sizes, accounting for that second layer of uncertainty is often helpful. And the final one's probably the most important thing for adaptive trials is it doesn't matter, the Bayesian quantities don't depend on the trial design. So if you haven't seen this example before, I don't know how many of you have, standard one, if somebody comes in and says, today I ran 40 experiments and 15 of them were successful. Some kind of dichotomous outcome. My data is 15 out of 40. And I just want to do some test. I want to know, is my proportion of successes equal to 0.5 or not? So if you go for a second, you go, okay, I'm going to quickly compute that, what the p-value is for that. There's a question you have to ask, which is, how do you design your experiment? If you designed your experiment where I'm going to do 40 experiments and I'm going to count the number of successes, you get one p-value. If you did an experiment where you said, I'm going to run till I get 15 successes, and then I'm going to stop, which is another possible reality, you get a different p-value. If the person tells you, oh, I just ran experiments until my partner said it was time to come home for dinner, then that's an entirely different experimental design. And to find the p-value, you have to have a lengthy discussion about dinner habits, which is a little awkward. So anyway, the Bayesian answer doesn't depend on any of those. So no matter how you did the experiment, the 15 out of 40, like those pieces of the likelihood decide the answer. And if I'm going to be doing adaptive trials, in an adaptive trial, there are interim analyses and the trial evolves over time. This is the desirable feature that at the end of the day, I will try not to break your place for it. But anyway, at the end of the day, it's good to have some consistency there. Okay, so let me kind of dive into the case studies here. So the first one is the COVID vaccine trial, which is a Bayesian adaptive trial. It's a pretty simple one. If you haven't run a vaccine trial before, vaccines are evaluated in terms of something called vaccine efficacy, which is the percentage of infections that are removed in the treatment group. So if 100 people were going to get infected in the control and you have 40 people get infected in the treatment, and you've gotten rid of 60 of those 100 infections, 60% vaccine efficacy. So that's the standard terminology there. You could define it differently. Typical vaccine trial, we enroll lots and lots and lots and lots of people. All of the COVID vaccine trials enrolled tens of thousands of individuals. They vaccinated them and then they followed them up over time. They followed them up over time for two purposes. One is safety. So a lot of bad things can happen in a vaccine in the first day or week or two after you get a vaccine. Those are all rare, but we want to know what they are. They also are interested in long-term follow-up. So if the side effect of the COVID vaccine is we all become zombies in 10 years, we don't know that yet. It would be a bad awakening for all of us. But in any case, the other thing, of course, they want to know is efficacy. So they follow up and count how many people got infected and whether that happened in the control group or the vaccine group. So the FDA at the time required that they were going to demonstrate superiority to 30%. So you didn't just have to beat the null. You basically had to be better than 30% vaccine efficacy to get approved. So it's a super superiority test. So that, of course, means that 60% is a good vaccine. And Pfizer actually thought that their vaccine might be at 90% based on their preclinical their early phase data that they had at the time, which of course was not a ton and anyway, not the best. But anyway, they had hopes. So this is that situation where I had the confidence interval from -1 to 5. Pfizer's in the same boat. 60% requires a very different sample size than 90%. So that's what led Pfizer to do something adaptive. I've talked about these kind of trade-offs. The notion is if you run a really large trial, it may just be too large and it may take extra time to get a vaccine to the public. If you run a small trial, you may be underpowered and not learn anything. Both of those are bad. So the notion is if the solution here is what I'm going to do is I'm going to let the data guide me to the right sample size. I'm going to start small. I'm going to do an interim analysis. I'm going to look and see are things going well. If they are, I'll declare success. If they're not, I will continue and let the trial get bigger. And one thing you should keep in mind is you should remember, well, if you look at the data twice, you're generating an extra type 1 error rate. Absolutely. So there's an adjustment where those are not p less than 0.025 looks. They're a little less than that to account for the multiplicity. All right, so this is a, I'm going to go through this one quickly. I do a lot more of this when I'm talking to clinicians. But anyway, this is what a dud vaccine looks like. So these are just sample paths. So all I did is I just simulated binomial data and I'm plotting what proportion of the infections that I observe are in the vaccine arm. So this is the number of events or infections. And then this is the proportion that are in the vaccine arm. Ideally, I would like the proportion in the vaccine arm to be 0, but of course that may not happen in practice, but the less in the vaccine arm, the better. This is the kind of pass that you get for a dud. So the usual stuff, you know, central limit theorem, we converge to whatever the truth is. That's what's happening there. This is what a 60% efficacy vaccine looks like. I'm sure this should all be, you know, standard stat 5s. What was the course called that I taught you?
601.
You still have the book?
I had the syllabus.
Okay.
Yeah.
All right. I don't remember that you did badly. I thought you did fine. So,
You're thinking the second time I took it.
may not have taught you the first. Maybe that was the reason. Anyway, all right. So Jeff was great. So you guys should understand. Anyway. All right. So you know, the usual thing to do with statistics is we find a point where these have separated enough. So we've got our 2.5% type 1 error above the line. We've got our 90% power below the line, and these don't separate enough until 150. And so this is where perhaps our fixed sample size would be, and this is our threshold for success. So we got to get out there. Okay, so the basic idea of all these adaptive trials is, do I have to wait that long? If I happen to get results that are here or here or here, those are pretty convincing too. None of the black curves are in that region. So a dud vaccine doesn't achieve results that are here or here or here with any kind of likelihood. So why can't I declare success there? And so this is what Pfizer did. So this is from their protocol on what their rules were. They put in interim analyses. The first one was meant to happen at 32 events, and they had certain splits that had to be achieved. So if those 32 events, if they had 6 in the vaccine arm and 26 in the control or better, they would say the drug works and they'd be done at 32 events. They also had a futility rule. If the split was actually going against them, they might stop for that. If you don't stop for success or futility, you just continue. So the next interim's at 62. The split has to be 1547 or better, and so on. Now this is, it is defined in terms of a Bayesian rule here. Probability the vaccine efficacy is greater than 30%. That had to be over a particular threshold. There's really nothing, the Bayesian is not doing a lot of work here. So you could easily do this as a standard alpha spin group sequential, and it would probably look very, very similar to this. So Bayesian is not doing anything, it's just an example of an adaptive trial. All right. So these are, to give you an idea about where these success criteria are, what Pfizer has effectively done is they've said if we can enter any of these red rectangles, we win. And so the idea is that all of those red rectangles are far below any of the null paths. And so this gives us a reasonable chance. And these red paths have been chosen so that the chance that any of the black paths will go in there at all is 2.5%. So even doing multiple looks, we've got our type 1 error control on here. So this is what was done in COVID. As it turned out, this trial actually did not get run according to plan. When it was designed, that was in the summer of 2020. There were relatively few infections. The trial got run in the fall and winter of 2020. The infection rates in the country skyrocketed. So as it turned out, they actually got tons and tons and tons of, not tons, but they got a lot of events in a hurry. So they ended up, instead of those interims at 32 and 64 and whatever, by the time they could get their lab to process the data, they had 90-something events. In addition, the 90-something events were way in their favor. It was 91% vaccine efficacy. I'm not going to get into little details. If you don't run a trial as designed, you have to go, okay, I need to think about that. Fortunately, this trial won by so much. All of the I have to think about that end up in the place where, of course, it works. So this was approved. It was given an emergency use authorization and then final approval next year. These kind of trials are done a lot. I don't know a lot of research in this area involving just a straight group sequential kind of design that's one-to-one randomized. There's some, but not as much. The kind of more interesting questions that people are worried about in these issues is what happens when the outcomes are significantly delayed. So how do I do things like this when it's a knee device and a knee device has a 2-year endpoint and I'm enrolling 100 patients a year? So at any given point, I got a whole bunch of people that are in their 2 years of follow-up. Then how do I do things like that? And so there are issues where can I impute the final outcomes. So if I know somebody's knee is doing well at 1 year, that's highly predictive their knee is going to still be doing well at 2. In the reverse situation, if I know they've had an adverse event at 1 year, they're already done. So their endpoint is determined to be a failure. So the question is, can I use those kind of endpoints? There's a lot of stuff going on that. There are lots of things being done right now about other factors being optimized at the same time. So for example, if there's a dosing regimen that's happening at the same time as this, I may run a trial that has 5 doses. And then at some point I run interim analyses, I cut it down to 1, and then I start doing this kind of group sequential. That entire process is still fairly active. Selecting a subpopulation of people while you're at it, all of those are active areas. There's some discussion about the estimators in these settings. Anytime I do this kind of cherry picking, where an interim analysis, I can catch random highs in an interim analysis. So usually the estimators that you get out of this are biased. And how biased they are really depends on how you did the interim analyses. You can do them stupidly and generate big biases. You can do them better and generate lower biases. But anyway, that's a lot of the frequentist work in here is how do we make these estimators. And I'm not going to get into the details of this, but if you've heard the phrase of sample size re-estimation, it's kind of like this room. There are group sequential people on one side, there's sample size. Are you like, you're Switzerland?
It depends on the context.
Okay.
That's really what it comes down to.
All right. So anyway, but there's an ongoing debate between group sequential like designs and sample size re-estimation things, which have a long history as well. I will go ahead at the risk of offending somebody in here, that I do think the group sequential are just going to win this debate. One of the reasons that I think they're going to win this debate is imagine if I have the question of, do I need a sample size of 300 or 500? Well, when do I want to make that decision? A sample size re-estimation often makes it at like 100 or 150 or 200. So you're making a decision far into the future. You're deciding 300 or 500 when you have 100 or 200. If you do a group sequential, it makes the decision as late as possible. So the decision between going to 300 or 500 is made no earlier than 300. And so it has to be a decision that's being made with more data. And I would actually be, I don't have any proof that that's what the world is going to end up with, but I will be terrified if the answer to that question is decisions are better when you have less data. I think we should all worry about our careers if that's everything. So I think we'll end up on the group sequential side, and there is a lot of the simulations that I've seen indicate that the group sequential like designs do better. All right, second case study. West African Ebola epidemic. We talked about at dinner how I almost got arrested in Canada over this. Anyway, so in Ebola, the primary endpoint is mortality. So we expect a 40% mortality rate from the disease, and you want to make that lower. So very clean endpoint. When the epidemic was going, there were multiple therapies proposed. Actually, something like 5 or 6 that were active. There were probably another dozen that were less enthusiasm, but some people liked. You could give these in combination as well. So why don't we give not just A, but we'll give A and E together, for example. And so this is, there were some rules on this. There were some restrictions on populations. Children and pregnant women couldn't take certain things, so on and so forth. I'm going to make this problem simpler for this discussion and say, suppose there were only 4 potential therapies, and here's the problem in front of you. I can give any of the 4 alone. I can give any of them in a combination of 2. So that's 10 possible arms: A alone, B alone, C alone, D alone, A and B, A and C. Fill out all the possibilities here. So now the question is, well, how do I investigate this? How do I investigate 10 possible arms? And so this puts me in the same situation of if I'm not adaptive, I suddenly have to take some risks. And so one of those paths that I could go forward is investigate one at a time. So let's pick A, and let's investigate that and run a trial. If it works, I will be very happy. If it doesn't work, I will go on to the next one. Maybe that's B. So Jeff, what would you pick first? C. C, okay. So anyway, Jeff would then pick. Your other question is, in addition to what order do you do them in, do you do the combinations first? Do I do, for example, A and B? And if that works, then I'll try A alone or B alone. Do I need A and B to work to try A and B together? Or anyway, how does that work? The basic problem with investigating one arm at a time is you're at the mercy of getting the right guess. So like if Jeff picks C, Jeff looks amazing if C is actually the one that works. On the other hand, what's the last one you would have picked? D. Okay, you would have gone through all the combinations and then worked your way back to D. Okay. Anyway, if D is the one that works, Jeff is the most horrible person on earth. So anyway, but it's high risk in that. The other thing that you could consider doing is investigating all the arms simultaneously. And this is also potentially bad. I've got 10 arms. So we had access to an Ebola clinic. This was a trial we designed for the Gates Foundation. So we had an Ebola clinic. We had a sample size of 250. So if we investigated all 10 arms simultaneously, we can do 25 patients per arm. That's not really enough to do. That's not a very highly powered study for any individual arm. So this also has problems. So the idea behind the adaptive trial is to try to bridge the gap. And sorry, I didn't say the advantage to investigating all arms simultaneously is if one of the arms works, you're investigating it on day 1. It's in the trial. So that's the key thing that has going for it. Okay, so we did something called response adaptive randomization, or we proposed. I'll get to what happened to this trial as well. Basically, all my case studies are going to involve just complete failures, except for the last one. So you should keep in mind that that probably isn't reasonable how clinical trials actually work. But anyway, response adaptive randomization. So we began randomizing to all 10 arms simultaneously. So every arm got a few patients. I think this number was actually, there were 3 assigned to each of the arms. If you're thinking 3 is not many, you are correct. We also had a model which it was an additive model for the combinations in here. So there's a linear model in here. So if I'm estimating the effect of B, for example, it's not just the people who only got B that contribute to that. It's the people who got B, who got A and B, who got B and C, who got B and D. So it's a little more than that, but it's not many people. And then what we did is we did an interim analysis. And we said, okay, which of these arms are doing well, which of them are doing poorly? The arms that are doing well, we're going to increase the allocation a little bit. The arms that are doing poorly, we're going to decrease the allocation a little bit. Then we're going to collect some more data and we're going to do it again, increase a little bit to everybody's doing well, decrease who's doing poorly. Your first thought here is, boy, that could be really stupid. And you're right, it could be. So the thing that you can do wrong in this case is be too aggressive. Arm A cured 3 out of 3 people. Hey, let's give arm A to 95% of the future patients. That's not very clever, because if arm A just got lucky, you've eliminated your ability to collect more information. So there's an interesting math problem here of, okay, how much do I increase these probabilities so that I get the right answer? And you don't want to increase and decrease them too quickly, or you will not get the right answer. So this is a key part of the theoretical discussion in here. If you do this right, however, the trial does eventually focus on the best arms. By the end of the trial is likely allocating 80–90% of the patients to the couple arms that are doing well. I'll show some examples of this in a second. And so the idea is this is bridging that gap by we're investigating everything at once. Remember, the problem with investigating everything at once was I didn't get the sample size on the arms. I'm doing that by trying to eliminate the ones that are doing badly and focus on the ones that are doing well. So by the end of the trial, I actually do have a manageable sample size. All right. So how well does this work? So this is a possible example. This is the table here is giving all of the possible combinations of the drugs. So this is A alone, B alone, C alone, D alone. This cell would be the combination of C and D together. So this is the 10 possibilities. The blue number in here is the underlying truth that we're trying to figure out, does the trial work? So our first question is, suppose D works. So there's a 40% mortality rate if you don't give D. There's a 20% mortality rate if you give D. The answer to this question is give D. We had a side discussion over would you rather give a combination or an individual therapy. At the time, the answer was, I don't care as long as it saves people's lives. We'll figure out whether the combination is needed or individual components later. So that was a conscious choice to do that. Okay, so next numbers here. How many patients get allocated to each of the arms? So this is through that process. In order to find these numbers, we do something called trial simulation. So basically we're doing, this is really just Monte Carlo integration. We simulate an entire trial through, see what the results would be, simulate another trial all the way through, simulate another trial all the way through, and so on. So basically for power, we're just counting how many of the trials were successful. It is purely Monte Carlo integration, just random generate from the distribution. Okay, so this is the red numbers here say that on average, we give 8 to 11 patients each of these 6 arms. The reason there's an 11 for the combinations is our model had a slight preference toward combinations as opposed to singles. So that was, I'm not getting into the details of that, that was done on purpose. If you'll look, the arms where D is given, we're averaging 43, 50, 49, 49. So we're giving almost 200 of our 250 patients arm D, which is actually the right one. So we're benefiting patients inside the trial by doing this. We're focusing on arm D, which is what we want to do. These sample sizes, I'm not saying they're huge, but they're bigger than 25. So we've doubled those. And then the last number here, this green number, this is the probability at the end of the trial I have to make a decision and I have to tell the world what's the best arm. And so these are the probabilities that I pick each of these arms. You can see for the 6 that don't work, the probability that I pick them is 0. I never make that wrong choice. And then these 21% chance only D, and kind of split evenly between these others. So basically there's 100% chance at the end of the trial, I will say, give D in some form. I'm getting the right answer here. A little bit more complicated scenario. This is a situation, arms A and B don't work, 40% mortality rates. Arm C drops it to 30. Arm D is still dropping it to 20, but there's also this, if you give C and D together, you can drop it all the way to 10. And so again, you can see the focus here. The trial allocates most of the patients to the better performing arms. They're not all on this sweet spot here, but I got 90 of them on there. And I got a lot of the other people to arms that, if not optimal, or at least doing something for the patient. And then finally, the probability that I pick the right arm here, this is, you can see 0% chance essentially that I pick arms A, B, and C. I've got some chance of picking these suboptimal D arms. And then I've got about a 90% chance that I actually do give the right answer. This is if we ran a fixed trial where we just gave 25 patients each, this number would be 80. So we have a better chance of treating patients in the trial well, and importantly, we have a better chance of giving the right answer to the world at the end of this trial.
How many times do you reassess the model in the sound design?
I was hoping you wouldn't ask me that. The first one is at 30. I'm gonna make a guess that it isn't gonna be that far off, but I think we did it basically every 30 patients. And you can have a debate on this about how often. I think this was done in 2016. I think the more recent papers say if you do something like start at 20% and do about every 10% thereafter, everything past that's marginal benefit. If you do 20 like 20, 40, 60, 80% of your data, you get a lot of the benefit. So anyway, it's usually in that kind of range. All right, so this trial was also never run. And it was published, but the reason it wasn't run is it took a long time to prepare to get everything right. And by the time it was done and ready to go, there was no more Ebola, which is good. And it was actually the most bizarre thing where we're watching the numbers go down, going, hey, we can't run our trial. But anyway, obviously that was a good thing for us, that Ebola disappeared. But this trial never actually happened. So we've made plans to react to the next epidemic faster.
If it never happened, what drug was there?
Sorry, what?
So you said a trial did not happen, but a drug was used to treat the
So there were, yeah, and I don't remember, there were other trials being run at the same time, and they did a single arm. So actually a reasonable, and I don't know what that drug is to answer the question you asked. A reasonable argument here is you did something complicated that could have worked, but never actually got into the field, and other people did something simpler and actually made it happen. So it's a very reasonable argument at this point. I do not know what the other drugs that came out of that are. I do know the vaccines that came a couple, 2-3 years later look to be effective. But I don't, this space, after this trial, I haven't been in the space very much. I'm not sure. This kind of allocation method, this moving things up and down, this is very controversial in the literature. There are lots of good papers here. Honestly, there's lots of bad papers here that have straw man arguments. I have kind of put this essentially as these have been the equivalent of, I want to prove that a scalpel is bad for surgery. I will stab a patient repeatedly with it. Go, the patient died. You should not do surgery. So there are good ways to use a scalpel and bad ways to use a scalpel. And you got to be careful in this area of whether they're comparing a good way to a good way or a good way to a bad way. I think one of the key issues here is the newer designs that are coming out have basically shown if I do just a regular arm dropping design, where I start all 10 of the arms, and I don't do any of this allocation manipulation, but instead I just say, you're doing badly, you're out, it works as well. And doing this kind of arm dropping design is simpler. So all the trials that I do right now, we no longer do that. We do the arm dropping stuff. And again, that's because it seems to work as well, and it's easier to do. Let me skip that. Okay, and the last case study example, let me check on time. I'm doing more than I anticipated, but I'm still good for time in terms of the hour. Okay, so Ebola, we didn't get the trial run. This is bad. The reason was it takes a long time to prepare an adaptive trial. This is something that's lots of moving parts. So what we did is we said, hey, we're never going to get caught in that boat again. So we then decided we're going to be ready for the next epidemic. We're going to plan the trial before the epidemic hits. And so we designed, we asked people, well, what was the most likely next epidemic? Everybody said some kind of pneumonia. And so we designed basically an upper respiratory epidemic trial. And we actually also designed what is called REMAP-CAP, which was a trial just for ordinary community-acquired pneumonia, which people have continuously. So REMAP-CAP is a trial that was running investigating pneumonia therapies. But REMAP-CAP also had a set of explicit break in case of pandemic chapters to the protocol. And so it said, if there's ever a pandemic, we are going to do the following, and all of the stuff that we had to do in Ebola while the epidemic was raging, we're all sitting in this prepackaged instructions. So obviously in March of 2020, there is such a pandemic. That sleeping stratum was open, and REMAP-CAP was enrolling patients by the end of March 2020. I will not say this was an incredibly smooth process. A lot of people lost a lot of sleep. There were tons of hiccups, data issues, all the stuff that you would expect to have happen in a clinical trial. I'm not going to say this is, but it was running in March of 2020. The other advantage to this is because it was piggybacked onto an existing CAP trial, all of the data infrastructure was already there, the randomization systems, all of that stuff, all the medical monitoring, it was already there and good to go. That part did not have to be built from scratch, except for particulars with COVID, but there was a lot of things that could be reused. REMAP-CAP is actually a more complex trial than the Ebola one. And so it's a platform trial. And the idea is also this problem of how do I investigate multiple therapies at once? But a platform trial, what it's aimed at is at the beginning, I might enroll a control and say 4 treatment arms. And a platform trial is set up to be perpetual. So I might do interim analyses and maybe, for example, treatment A stops for futility. So it doesn't look like it's going to go well. At that point, treatment A would disappear and it would get replaced with treatment E. Next trial that goes out, treatment F comes in, and so on. So the idea is that I can keep replacing the arms in the trial, but I maintain the entire infrastructure. So this has been kind of a major area of research focus these days on how these trials work. Happy to answer tons of questions about them. But anyway, the idea is essentially threefold. One is we're sharing the control patients. If you're working this area, you should immediately think, oh, if you save control patients, you've got correlated type 1 errors and all the other kinds of stuff. There's lots of research going on in these kind of questions, but it does save sample size. All of the stuff that the Pfizer vaccine trial did in terms of adaptive stopping, that's all going on here. And investing the saved resources forward into new arms also is some advantages. Let me kind of skip to the main course here. An idea about how efficient this is. So suppose that I just ran, I need to investigate a lot of arms. And I'm going to run one arm at a time. So I'm going to do what Jeff says, I'm going to run arm C. Did arm C work? If yes, I go, woo. If no, I go on to the next arm. I do that over and over again. You could ask the question, how many treatments do I have to explore before I find something that works? And obviously this depends on what is the likelihood that a treatment works. So all of these are done under the assumption that there's a 10% chance any individual drug you investigate works. So if you do separate trials, the expected end is about 1966. If you just share the controls, so that means you investigate 4 arms at a time, then you can drop that. So you don't have to enroll as many controls, drop it to 1528. If you add the adaptive features, so this is if you do the COVID stuff from the Pfizer COVID vaccine trial, this drops it to 971. And finally, if you start reinvesting these resources forward, every time something goes out, something new comes in, this drops it to 849 for equivalent power in type 1 error. So this is not, a lot of times people work on, hey, I did this, I got 1% more power out of the design. This is 50% sample size savings as a society. In addition, REMAP-CAP has a factorial structure. So it doesn't just explore one set of arms, it explores multiple what are called domains, which essentially every statistician would call a factorial design. And I'm not really sure the history of how this happened. I'm very upset that it did. But anyway, so for example, REMAP-CAP might be exploring steroids, multiple ways of giving steroids, multiple ways of putting people on a ventilator, multiple ways of giving people antibiotics, and it explores all the combinations of those. And the underlying model doesn't look any different than what you do in a factorial experiment now. So you have your standard linear models that work in order to do this. At each of the interims, I need to close this off. REMAP-CAP can drop an arm within a domain. We might say, hey, this ventilator strategy doesn't work, you're out. We might declare an individual arm within a domain the best. We might add a new arm to a domain. We might add new domains. As is, and I am running a little bit of time, so I'm skipping a little bit here. But REMAP-CAP has enrolled 14,000 patients with COVID. It has done 66 different domains. Or sorry, 66 arms in 18 domains. So it's done a lot of the standard of care for COVID these days comes from a number of platform trials, but REMAP-CAP is one of them. I don't want to say that adaptive trials are fantastic all the time. As I said, they're a lot of work. We didn't get the Ebola trial to actually go in the first place. They require a lot of expertise to implement. You need people who can maintain a database and keep it reasonably current and accurate in order to do these interim analyses. You need people who are used to looking at data in this form and knowing the kind of quirks that can happen with it. So this is a lot of training in order to make this kind of stuff happen. It's still viewed as less standard by regulators, but they do take it. Certainly there's a guidance document in every major regulatory agency for how to do adaptive designs and a path to get things accepted. And let me stop there.